This presentation that I'm going to talk about today regards basically rectal cancer and new insights on some parameters and some factors, mainly tumor-associated neutrophils, Arginase-1 and Perineural invasion. Why rectal cancer? Well, rectal cancer is one of the most common types of cancer, being under the umbrella term colorectal cancer. Then whether this umbrella term of CRC, colorectal cancer, is still valid nowadays, I think that's also an interesting topic of discussion due to the fact that the clinical knowledge that we have and scientific knowledge that we have about colorectal cancer indicates how outdated CRC as an umbrella terminology is. Right colon cancer is very different from left colon cancer and very different from rectal cancer. So a need for an update on this terminology, I think it's due. This study focused on is an analytical study conducted between the Olimpia Medical Center, of which I'm the healthcare director, and the University Hospital of Besançon in France, and the Regional Tumor Bank of France Comté, which I thank for the help in conducting the basic research of this project. When I was in medical school, my thesis was on a discovery I made regarding the association between tumor-associated neutrophils and rectal cancer. These neutrophils being and having both a prognostic and a predictive impact. This study that we conducted this year is a very focused study, as we were able to gather insights on some factors that were not previously studied, especially in rectal cancer, namely Arginase-1 and the impact of Perineural invasion on this pathology. Why colorectal cancer again is because it's becoming an oncological pathology with both an increase of incidence in younger people and an increased rate of mortality in these young patients, even though, of course, with all the prevention campaigns in developed countries, diagnosis and treatment of colorectal cancer has indeed improved. The histology that we're going to discuss here is adenocarcinoma of the colon, and in this study we evaluated samples from 65 patients, on which on these samples we did two different stainings. We did an CD66B staining for the tumor-associated neutrophils quantification, and an Arginase-1 staining for the quantification of Arginase-1 levels. The interesting part also is during the analytical phase of the study, where different factors have been taken into account and have been removed during the analytical part of the study. For instance, as we will see in the results of this analytical part of the study, Perineural invasion has been shown to have quite an impact on both Arginase level and prognosis in this retrospective study. Why did I decide also to focus on Perineural invasion? Because Perineural invasion has been in the literature and in the clinics shown to be a negative prognostic feature in terms of rectal cancer. The reason why this could be can be different and various. For instance, the Schwann cell interaction with the tumor, including the recalling of MDSC, myeloid-derived suppressor cells, which act as immunosuppressor, increasing production, for instance, of TGF-beta, okay, and then creating an immunosuppressive environment in the tumor microenvironment. So you will see that in this analysis, different parameters have been taken in consideration. So regarding some of the results, here we see an Arginase-1 staining in, so here perineural invasion has been removed, so only patients without perineural invasions, where patients without neoadjuvant therapy had a significant, statistically significant higher level of Arginase-1 within the tumor. When I speak about within the tumor, I mean either in the deep core of the sample or in the invasive margin of the sample. Here we see that the same type of analysis has been done for the CD66B stain, meaning the tumor-associated neutrophils quantification within the tumor, where we see that those who didn't have a neoadjuvant therapy had a higher level of neutrophils. So this increase in neutrophils within the tumor was directly proportionate with an increase in Arginase-1 level. Here we see again Arginase-1 staining. However, this was in the deep core. Again, the increase in Arginase-1 is seen both in the deep core of the sample of the tumor and in the invasive margin. When patients didn't have neoadjuvant therapy. When I speak about neoadjuvant therapy, I speak both of radiotherapy and chemotherapy combined, or chemotherapy alone. Most common regimens, just as a side note, most common regimens of chemotherapy in colorectal cancer treatment, stage 3 colorectal cancer treatment, is either FOLFOX or FOLFIRI, combination of therapies involving 5-fluorouracil. Here we have again CD66B staining, but this time in the deep core, okay, and we see again that even in the deep core we have higher level of neutrophils in patients who did not receive or who did not undergo any neoadjuvant therapy. Then here is an interesting figure, figure 9, because in this scatter dot plot graph, we see how this is a comparison of Arginase-1 levels simply between patients who had perineural invasions and patients who did not have perineural invasions. And we see that when we take in consideration the whole group, but with the but when comparing the perineural invasion patients, we see how Arginase-1 is higher in the group with perineural invasion. Okay, this again may be possibly reflecting the let's say etiopathological origin of the immunosuppression in the tumor microenvironment caused by this neural tumor interaction, this Schwann cell tumor interaction, maybe, or a general neural tumor interaction, such as mentioned before, for instance, with recalling of myeloid-derived suppressor cells. Here again, we see, but with the neoadjuvant treated group, okay, of patients, where we see how the therapy acts on this relationship between Arginase-1 level and perineural invasions. in fact, here, where the patients were actually, we selected the patients treated by neoadjuvant therapy, we see how this relationship is basically inverted, because we see how patients without perineural invasion have a higher Arginase-1 level. Okay, so just focusing on patients treated with neoadjuvant therapy. And here we see another two interesting graphs. Basically, here we have the both in Figure 11 and Figure 12. We have both the quantifications of Arginase-1 level. Okay, and we see the division between the different neoadjuvant therapies. Basically, chemotherapy, no neoadjuvant therapy, radiotherapy, and radiochemotherapy. And we see how Arginase-1 level is much statistically significantly higher in the group that did not have any neoadjuvant treatment. Okay, and so this is also very interesting to note how the therapy itself acts on within the tumor microenvironment and Arginase-1 level. And here we have a couple of, let's say, the disease-free survival curves, okay, that are highly interested. First, Figure 13, we see both groups without perineural invasion, okay, and as we maybe imagined, the group with neoadjuvant therapy had a statistically significant improvement in survival. Here we did, what I did was simply a division of the different neoadjuvant regimens, where only the group who underwent radiotherapy, so the purple line, had a statistically significant improvement in DFS compared to the rest. Why only these two curves, survival curves? Because these ones were the ones that came out statistically significant and were of interest, as you will notice. So how perineural invasion really had, in this sample group, had quite an impact in terms of the analysis. Okay. So now, what does all this mean in terms of new insights and reflections on this data and on rectal cancer, Arginase-1, and perineural invasion and neutrophils? So first of all, that literature showed us how Arginase-1 seems to be a worsening factor in terms of predictivity and prognosis. It has been found in literature to be a potent immunodepressor. Okay, for instance, give an example of T-cell immunodepression, and arginase inhibitors are currently being thought as a, let's say, potential category of drugs to be used in cancer immunotherapies or as a therapy against cancer. In fact, inhibiting arginase in literature showed a strong boosting of immune reactions against cancer. Secondly, secondly, this relationship between high neutrophils and high arginase, so in this immunodepressive environment, may be related to a higher level of N2 neutrophils population within the tumor. As we may know, neutrophils, for instance, like macrophages, have polarization, N1 being more anti-tumoral and N2 being pro-tumoral immunosuppression and this type of features. And this data can possibly convey a fil rouge between perineural invasion and immunosuppression of the tumor microenvironment, higher population of N2 neutrophils within the patients who did not receive neoadjuvant therapy or who had perineural invasion, and a higher level of Arginase-1 in these patients who have higher levels of possibly N2 phenotype of neutrophils, which could be an interesting insight for future research in terms of further analyzing this field for different reasons, to develop prognostic factors or scores in an anatomopathologic setting, but also for development or contribution for new therapies such as arginase inhibitors. And I think also maybe the importance of analysis and studies like this one is to understand better, to gain more insights on the etiopathogenesis of cancer and the role of the innate immune system, both in terms of the cells of the innate immune system, but also of the different components, such as, for instance, Arginase-1 in cancer development and progression. And so, yes, just in order to conclude, Arginase-1 does seem to be a worsening factor to prognosis, and I hope that with this study I was able to shed new light on these novel relationships that could help this field in developing further research in this topic, and with the ultimate goal to aid patient care and treatment. Thank you for your attention.